Hello everyone, I am Mariela M. Arden, and in this presentation, we'll be diving into the world of Leishmania tropica. Let's uncover the basics of this tiny parasite and its impact on us. But before we dive into the details, I'd like to provide you with a roadmap of what we'll cover today. Firstly, we have the description of Leishmania tropica and its morphology, habitat, distribution, life cycle, pathogenesis, diagnosis, immunity, treatment, then prevention and control. Now that we have a roadmap of what lies ahead, let's dive right into our discussion. Leishmania tropica. Leishmania tropica is a protozoan parasite that causes cutaneous leishmaniasis, which is a disease transmitted to humans through the bite of infected sandflies. It is a tiny, one-celled organism with a tail-like structure called a flagellum and a part called kinetoplast near its back end, like what you see here in the picture. Next, we have morphology. Leishmania tropica has two forms: premastigotes in the sandfly vector, which are elongated and motile, and the mastigotes within the host cells, which are smaller, non-motile, and no flagellum. In other words, premastigotes are long, slender, and spindle-shaped, and have a single nucleus that is centrally located, and also kinetoplast located transversally near the anterior end. While the mastigotes are small, round to oval bodies, resides inside the monocytes, endothelial cells, polymorphonuclear leukocytes of the host, and the nucleus is large and lies at a right angle to the kinetoplast, which is slender rod shaped. Habitat. The primary habitat of Leishmania tropica is within the digestive system of the female sandfly, particularly species of the Phlebotomus genus, which serve as vectors for transmission. Within the sandfly, the parasite undergoes several developmental stages, including multiplication and differentiation, eventually leading to the infectious form. The secondary habitat is the mammalian host, including humans. Once transmitted to a human host through the bite of an infected sandfly, the parasite can inhabit various tissues, particularly the skin, where it causes cutaneous lesions characteristic of cutaneous leishmaniasis. Now, let's proceed to the distribution. As you can see here in the picture, there are different colors here because leishmaniasis also comes in different forms, which are cutaneous and visceral. Also, there are different parasites that cause them. However, for now, we'll be focusing on cutaneous leishmaniasis, which is represented by the yellow-green color here. Leishmania tropica is one of the parasites responsible for cutaneous leishmaniasis, and it is prevalent in the Mediterranean basin, the Middle East, North Africa, and parts of Asia. Next, we have here the life cycle. The life cycle of Leishmania tropica involves two hosts, the sandfly vector and the mammalian host. The parasite is transmitted when an infected female sandfly takes a blood meal from a host, introducing premastigotes into the host skin. Inside the host phagocytes, premastigotes transform into amastigotes, multiply, and eventually rupture the host cell, releasing new amastigotes to infect other cells. When the sandfly feeds again, it ingests these amastigotes, which then transform into premastigotes in the sandfly's gut, completing the cycle. Leishmania tropica is primarily the cause of cutaneous leishmaniasis, leading to skin ulcers and scarring, which are the symptoms of it. The parasite infects macrophages and other phagocytes, impairing their function and causing local tissue damage. Cutaneous leishmaniasis shows up as painless skin ulcers with raised, irregular edges. These ulcers can take months to heal and may leave scars, like here in the pictures. Our body's defense system is crucial in fighting Leishmania infection, a type of immune response called T helper 1 cells, and the production of certain proteins called cytokines help control the infection. However, a strong immune response can also harm tissues. To find out if someone has Leishmania tropica infection, doctors use a mix of methods: clinical examination, serological tests, and microbiological methods. Clinical examination helps identify characteristic skin lesions, while serological tests, such as the direct agglutination test or enzyme linked immunosorbent assay, detect antibodies against the parasite. Microbiological methods, including culture, polymerase chain reaction, and antigen detection, provide direct evidence of the presence of the parasite. The treatment for cutaneous leishmaniasis caused by Leishmania tropica usually involves antimonial based compounds, such as sodium stibogluconate or meglumine antimonate. Alternative treatments include amphotericin B, paromomycin, and multifocin. The choice of treatment depends on factors like the severity of the disease, the patient's age, and general health, and also the potential for drug resistance. Preventing and controlling Leishmania tropica infection primarily involves reducing exposure to sandfly vectors and improving the living conditions in endemic areas. Some preventive measures include using insect repellents, wearing protective clothing, sleeping under insecticide treated bed nets, and controlling sandfly breeding sites. Vaccination is not currently available for leishmaniasis, but research is ongoing to develop effective vaccines. And that's all for our topic, Leishmania tropica. Thank you all for your attention, and with that, I close the presentation. Thank you.